Imagine your brain is this incredibly complex, constantly buzzing electrical grid. Right now, you've got billions of neurons rapidly firing off action potentials, sending critical messages back and forth just to keep you functioning. And we spend a lot of time thinking about how these neurons fire, right? How the spark is lit. But I want you to consider something that is arguably way more important. What exactly prevents those highly active, rapidly firing electrical signals from just running completely out of control and frying the entire system?
Welcome to This Explainer. Today, we're going to explore what I really think is the hidden hero of neuroscience. It's a phase of the neural cycle known as after hyperpolarization. It's a bit of a mouthful, but it essentially acts as the brain's ultimate built-in braking system.
Here's our roadmap for today. We'll cover 1. The brain's braking system, 2. Defining the undershoot, 3. Fast, medium, and slow, 4. Preventing neural overload, and 5. Limits of the brakes. Let's just jump right into it.
Section 1. The brain's braking system. So, let's start by really understanding the fundamental biological problem we're dealing with here. Neurons are incredibly excitable and active by design, but without a strict, highly tuned regulator, they would just send chaotic, runaway signals. They absolutely need a way to stop. To figure out where this stop actually happens, let's just trace the exact timeline of a single action potential. A neuron sits at a resting phase, right? Then it spikes up during the rising phase as it depolarizes, and then the voltage crashes back down during the falling phase. But I want to hit pause right on step 4, the undershoot, because this brief little window is where the regulatory magic actually happens.
Section 2. Defining the undershoot. Now that we've pinpointed it in our timeline, let's establish exactly what this phase is, both chemically and functionally. So, in neuroscience, this undershoot is formally called after hyperpolarization, or AHP, and it is so vital to understand that AHP isn't just some passive reset where the neuron is, simply catching its breath. It's a highly active phase. It's kind of like slamming your foot on the brake pedal of a car so hard that you actually roll backward a few inches. The voltage of the cell drops below the normal resting baseline, going from around -70 millivolts down to roughly -75 millivolts. By driving the voltage down so low, the cell is actively putting the brakes on the neuron, making it much harder for it to accidentally fire again.
And as the source material brilliantly points out, this sharp drop creates a refractory period within the sodium channels. Honestly, this is an absolute stroke of evolutionary genius. This braking period essentially closes an inactivation gate, completely eliminating the possibility of an action potential moving backward. It ensures that electrical signals travel strictly in one direction down the axon. Section 3. Fast, medium, and slow. Because it turns out, this braking system isn't just a simple on and off switch. It actually comes in 3 distinct flavors.
Taking a step back to look at the big picture, these 3 types scale dramatically in duration. We're talking anywhere from just 1 single millisecond to over a full second. And what's really amazing here is they rely on completely different biological hardware, specifically different potassium channels, to perform highly specialized jobs for the neuron. Comparing the extreme ends of the spectrum really highlights the contrast. The fast AHP lasts just 1 to 5 milliseconds. Think of it like the anti-lock brakes on your car, rapidly tapping to allow fast-firing neurons to maintain ultra-precision spike timing. But on the totally opposite end, the slow AHP can last for seconds at a time. It acts as the emergency brake. It steps in after a burst of heavy activity to completely stop repetitive, runaway firing and essentially let the whole system cool down. And hey, we definitely can't forget the medium AHP, sitting right there in the middle. If fast is anti-lock and slow is the emergency brake, well, the medium AHP is the brain's cruise control. Lasting anywhere from 10 to 100 milliseconds, it's responsible for perfectly pacing the firing frequency, setting the exact interval between spikes. Interestingly, it also plays a role in suppressing long-term potentiation, keeping overall neuroexcitability perfectly in check.
Section 4. Preventing neural overload. Okay, so how exactly does the brain recognize when it's time to yank that emergency brake?
Well, the slow AHP doesn't just happen magically on its own. It actually requires a pretty complex indirect signaling pathway. When a neuron fires in heavy bursts, calcium starts building up in the cell body. Then, proteins with, frankly, wild names like hippocalcin act as these specialized sensors. They detect this calcium spike and trigger a secondary chemical cascade that releases a molecular messenger called PIP2. It's like an incredibly complex chemical relay race that eventually activates the potassium channels. This indirect route is exactly why the slow AHP has a delayed onset, but also why it provides such a long-lasting, powerful shield against excessive neural discharge.
But wait, alongside the AHP channels, the cell actually has another trick up its sleeve, the sodium-potassium pump. This electrogenic pump works relentlessly to restore the ion balance after a cell fires. But crucially, as it pumps out sodium and pulls in potassium, it generates its own hyperpolarizing current. Meaning, the faster a cell fires, the harder the pump works, and the stronger this hyperpolarizing silencing current becomes. It's just a beautifully designed negative feedback loop built right into your own biology that actively shuts down overactivation.
Section 5. Limits of the brakes. Because as amazing as all this is, biology isn't perfect. Let's take it out of the petri dish for a second and into the wild to see what happens when these hyperpolarizing currents are pushed to their absolute physical limits. To see this in action, we can look at the weakly electric fish, E. virescens. It navigates and communicates by emitting a constant electric field from specialized cells called electrocytes. Now, during dominance fights, or when avoiding predators, the fish communicates by momentarily hitting the brakes on its electrical discharge. It creates this tiny pause in the signal, a submissive signal known as a chirp.
But here is where the system gets heavily tested. If the fish creates these chirps too often or for too long, the overall firing rate of the cell drops. And because the cell isn't firing as much, that sodium-potassium pump we just talked about, it starts to slow down. As the pump slows down, that crucial hyperpolarizing braking current progressively weakens. Eventually, the brakes essentially just fail. Without that negative current holding things in check, the cells become highly excitable, firing spontaneously and chaotically. The fish completely loses the ability to properly send its electrical signals. It's a total system breakdown.
Which brings us to a really fascinating conclusion. The electric fish shows us that while this hyperpolarizing braking system is a literal marvel of evolutionary engineering, it has strict physical limits. It requires a perfect balance of ions, precise timing, and carefully managed energy demands. So, it leaves us with a pretty compelling question to consider. If the precise neural brakes in our own heads have such strict biological limits, how close are we pushing our own brains to the edge of overexcitation every single day? Thanks for joining me on this explainer, and keep questioning the incredible mechanics happening inside your own mind.